So a lot of questions in the exams ask you about Meselson and Stahl's experiment. The idea of their experiment, is that they wanted to demonstrate whether DNA is replicated using conservative replication or semi-conservative replication. So there were two, before we knew this for sure, there were two ways that, DNA could potentially have been replicated. Either they would take the two strands apart and a new strand would line up on either side, and those would be your daughter strands. Or the other option was it would come apart, they would copy both sides, and then the new strands would both go together and the old strands would join back together. Okay, we now know that it is semi-conservative replication, so a strand of DNA will separate and you'll get a new one on either side, and then those two will become the daughter strands. But this is how we learned it, this is how we worked that out. Meselson and Stahl's experiment started off with DNA, double-stranded DNA, bacterial DNA, which was grown in a culture medium which had only heavy nitrogen. So in other words, it only contained nitrogen atoms which had one extra neutron. And if you know, think back to your chemistry classes from IGCSEs, that would make them weigh ever so slightly more. It wouldn't make any difference to the structure of the DNA. This is not the kind of isotope that is radioactive. It's not the kind of isotope that would cause any other issues. It's just ever so slightly heavier. So you'll often see diagrams or you'll see the results. They can demonstrate that this was heavy DNA because when they put it into a test tube and they spun it in a centrifuge, I'm going to see if I can hold that up there so that it can be a little bit more clear instead of against the background. Because it was heavy, it lay and made bands somewhere around there on the test tube. Okay, so when you see the demonstration, you'll often see the test tube with the DNA near to the bottom, and that's because it is heavy DNA. It wouldn't be really noticeable to us. It wouldn't be noticeable to the naked eye or any kind of feeling. You wouldn't be able to feel that it's heavier, but it is just heavy enough that it means it goes a little bit further when you spin it in a centrifuge. Okay, and it's just because each nitrogen atom in the DNA, and there's quite a few of those, each nitrogen atom contains one extra neutron, making it, I think it's nitrogen 15 instead of nitrogen 14. No other difference. Okay, so that's what they start off with, and they make sure that they only have this kind of DNA. So this is the heavy DNA. Then they take it out of its normal heavy DNA, heavy nitrogen culture medium, and in the first phase of replication, they only put it into a light nitrogen culture medium. In other words, all of the nitrogen, all of the bases that will now, from now on, be available to it are going to contain only light DNA, only N14 instead of N15. So, if you had just DNA containing this stuff, it would not go quite so far down. It would settle up here on your centrifuge as opposed to down here on your centrifuge. Okay, so what they did, starting off with this from round one, they then put it into the culture medium with the heavy DNA, and what happened is that the DNA separated and it formed, due to complementary base pairing, formed double-stranded DNA. It formed the daughter strands of DNA now with one heavy and one light molecule of DNA. And the way they demonstrated that was they then spun the samples in the centrifuge again. And now in the centrifuge, when they spin it in the centrifuge, it doesn't separate the DNA or anything. So this is now half heavy, half light. And what that does is it ends up making a band in the middle of the test tube. It's not like heavy DNA, which ended up at the bottom of the test tube. And it's not like light DNA, which all ends up at the top of the test tube. This is what we called hybrid DNA. So this is evidence that DNA replicates due to semi-conservative replication. Okay, you start off with your completely heavy DNA, it splits up and both of those strands get a new strand attached to them, and they now go off onto the daughter cells and continue on with their life. It's not like they copy themselves and then say, okay, go off and be free now. We are going to go back together because this is where we were always with. That's not how that works. So semi-conservative replication is you end up with the old strand and a new strand together. Okay, and they call them hybrid DNA because it is half heavy and half light. So you'll see in sort of stage 2 of the Meselson and Stahl experiment, you will get a band forming in the middle. And as long as you're often asked to draw them, as long as you draw it somewhere obviously in the middle, you'll be fine. Now, a lot of the time, these have a second, another round of replication with them. So you've now got your 2 strands of DNA. And they say, oh, what if we left them another generation in the light DNA? And what would happen there, again, is exactly the same. You would take apart the 2 strands and both of them would join together with a new strand, again, just made out of light DNA. Okay, so there's one. Now, keep in mind that last time we took that one apart and it was already blue. So now we have now got a double strand of DNA, which is now completely made up of light DNA. Okay, and because there were 2 of them, I'm gonna go for a bit of continuity here. So we have now got 4 strands of DNA. 2 of them are light and 2 of them are hybrid. Okay, and so what you'll end up with is those 2 hybrid DNA strands will go to the middle of your centrifuge test tube. I'm running out of hands here. I need an extra pair. And your 2 your 2 light strands of DNA will settle near the top. Okay, and so you can see you will have some light DNA band there and a hybrid DNA band there. And again, the whole idea of this is just evidence of how we find out the things that we now take for granted. I know that it does make sense that, of course, that's how it should work. You know, the old DNA, of course, it wouldn't like just copy itself and then go back together again. But it's not obvious if you think about other things in biology. You know, it's not obvious from the point of view of formation of RNA, because that is what happens when we form RNA. You will peel the DNA apart, the RNA nucleotides will line up along the template strand, and then they will peel away and go off to do the next stage, the translation stage. And then, of course, the DNA strand will join back up together. I know it's not the same thing, but the point is that we have to make sure that we understand how and why these processes work the way they do, so that we can go to the next step with them, so we can work out what happens from there, so we can work out how mutations happen, how they're detected, and so on and so forth. So that's the Meselson and Stahl experiment. It's one of the very few experiments that you actually do have to understand, but even then, keep in mind that they will often make ever so slight changes when they ask you questions about them in the exam. So for example, my example was that it might start with heavy DNA. It would be put through several generations in heavy nitrogen only medium. It's quite possible that you would get a question where the opposite is true. They might test it instead and they do several generations with light DNA medium and then put it into a heavy DNA medium. Exactly the same thing happens. Just be careful to read the question really, really clearly and make sure you figure it out as you're going through. So step by step, what should be happening next, and what results should I be getting from this? So hopefully that makes sense then.